Okay, so I'm going to talk to you today about the work I've been doing over many years on trying to understand the genetic basis of single gene disorders and monogenic skin disease. So over the years, I've seen DNA technology change dramatically. So when I first started, we had to amplify up each exon one at a time. But now, with the new high throughput sequencing, which was developed maybe 10, 12 years ago, we can now sequence an individual's genome very easily and very cheaply. So my time in the lab has changed dramatically. Rather than spending months or maybe even years trying to find out what is causing a particular disease, now we can do it relatively quickly. It's important for people with rare skin disease and rare disease to understand the genetic cause of their condition. It helps them understand. And what we try and do now, once we've identified the disease gene, is try and understand the actual mechanisms that be underlying that disease. And hopefully we can perhaps improve and develop new therapeutics that can help people live with these genetic conditions. So this is the strategy we have in the lab to understand monogenic skin disease. As I say, we always start off with understanding the genetic basis. So at the moment it's fairly routine stuff. We send the DNA sample off and it goes to whole genome or exome sequencing. And then there's a filtering system which then allows you to home in and identify the mutation that's causing the disease. And what I'm going to talk to you today about is what you do next. It's the functional studies to understand what the protein that's encoded by the defective gene, what it does in normal skin function, and to understand how the mutation underlies disease. So the focus is going to be on, as a disease example, is the Polyplanticratodermas. So these are there's a whole range of these disorders, and the primary common aspect is that they have hyperkeratosis, thickening of the skin, primarily on the soles of their feet and the palms of their hands. The presentation of the keratoderma can be quite variable, and to date there's 40 plus known disease genes that can cause the Polyplanticratodermas. Their functions have been linked to desmosomal biology, which is involved in cell adhesion, keratin biology, which is the intermediate filaments, and also in roles in signaling and stuff. Now, another interesting aspect of the keratodermas is that sometimes they don't appear in isolation, so they can be associated with other conditions, such as hair phenotypes, but also conditions like hearing loss, cardiomyopathy, and also cancer of the esophagus. So because we're studying a skin disease, it's a relatively accessible tissue. And so we have quite a number of resources and also strategies to understand disease mechanisms. So as I say, we can access patient biopsies. And this is incredibly useful because not only can we do immunohistochemistry, for example, on the skin biopsies and look at the histology, we can also take away the keratinocytes from these skin biopsies and make immortalized cell lines. So we have keratinocyte cell lines in the lab, which have different mutations associated with keratodermas in. We can also manipulate these cell lines, and we can knock genes out, for example, with small interfering RNA, or we can overexpress our genes to understand what they're doing. And of course, we can now use the fairly recently identified CRISPR-Cas9 genome editing strategy to knock in particular gene mutations or knock genes out. The other beauty about the skin is, as well as looking at the cells in monolayer, we can make them into 3D structures. So we can actually grow skin in the lab, and so it can mimic the patient's skin. And the beauty about these 3D skin models is, as well, is that you can then manipulate them. You can test drugs, or you can knock genes out to see if that improves the phenotype of the skin culture. So I think Professor Adele O'Toole will talk a bit more about this in her talk. And then, of course, there is some utility of looking at animal models of disease. So even though we can grow 3D skin models, sometimes it's good to have the organism, because there's often a crosstalk between different cell types. So it's not necessarily a disorder of the keratinocyte. There can be an interplay with various immune cells, for example. And of course, relevant to this course, with all this tissue and resource, we can then do omics. So you'll hear a lot about RNA-seq, but equally as other types of omics one could do proteomics and lipidomics to understand exactly how this mutation is affecting the palmar-plantar epidermis. So as I say, I'm going to go through a series of examples to show and to link the various techniques to that. So the first one I'll talk about is diffuse non-epidermolytic palmar-plantar So a striking phenotype associated with this condition is if the individuals immerse their hands or feet in water for only a short period of time, you get this white scaly appearance, as though you've been in the bath for about 2 hours. And we've identified that mutations in the gene that makes encodes a water channel protein called acroporin 5 is the cause of this condition. So acroporin 5 is a cell membrane pore protein that sits in the membrane and allows the osmotic movement of water across the cell membrane. Now, at the time of discovering the link between acroporin 5 mutations and this skin disease, we submitted the paper to review, and one of the reviewers said, "We don't believe this can cause this disease because acroporin 5 is not expressed in the skin. It is only expressed in the sweat gland." And to be fair, the reviewer was true in a way, because most studies in the literature just focused on looking at the skin from non-palmar plantar, i.e. the interfollicular skin. And I agree with the reviewer, acroporin 5 is not expressed in interfollicular skin, but it is expressed in the palm and sole, which is where the disease manifests. And you can see here in control skin that acroporin 5 is expressed at the cell membrane in the upper layer of the epidermis in the stratum granulosum. So what about the effect of these missense mutations in acroporin 5? What are they doing? Well, they're not affecting the protein. If anything, there's actually pretty more acroporin 5 protein expressed, with a lot of it at the cell surface. So it's as though the protein, maybe the mutations are stabilizing the protein in some way. So a variety of different missense mutations have been identified, and here you can see how they map across the protein. They're all over the place. It's very difficult to understand what these mutations may be doing to the channel. However, the crystal structure of acroporin 1, a family member, is known, and it shows some similarities to acroporin 5. What happens when you then sort of map these missense mutations onto this channel? You can see that all the mutations are lining the channel, the pore. And what they seem to be doing, and some of them are right in the pore, and what they seem to be doing is slightly distorting the pore, so it's leaking. So the mutations are causing leaky channels, and of course that fits in very nicely with the phenotype we see from the patient. I'm going to switch to another condition associated, Polyplanticratoderma associated with gap junction mutations. And the example I'm going to show you here is the mutations in a gene called connexin 26. So gap junctions are intercellular channels that in the skin allow the movement of small ions and metabolites directly from the cytoplasm of one cell into the other. So it's involved in intercellular communication. What we see is striking genotype phenotypes correlations with different mutations in this connexin. So the top panel is showing a patient with a condition called Faux-Winkle's syndrome. So as well as getting the Polyplanticratoderma, you also get these star-like keratoses on the ankles, and you can get constrictions around the digits, which can lead to autoamputation. This condition is always associated with a particular missense mutation called D66H. As well as having the keratoderma, this is also associated with a mild sensorineural hearing loss. Just a few residues down along the gene, we see another missense mutation, R75W. You can see the PPK presents differently to that of the D66H mutation. And the other thing I point out is that this is associated with a profound hearing loss. So different mutations in the same gene can cause different phenotypes. To add further complexity, There's a couple of other mutations. The one's called D50N, which is associated with KID syndrome. And there's another 2 mutations which also cause KID syndrome. And this is this is called keratitis, ichthyosis, and deafness. So it's a much more severe and generalized skin condition, which is associated with profound hearing loss, but can also lead to a development of squamous cell carcinoma. And yet, there's other mutations. This one here just causes hearing loss, no skin manifestations at all. Connexin 26 mutations are the major cause of recessive hearing loss, and there are many of those mutations. And these obviously just present with hearing loss with no skin condition. So you can see that there's a lot of genotype phenotype correlations going on here. Similarly, another connexin 31, again, has different mutations that present with different diseases. Now, most missense mutations, and these are 2 examples here, associate with a dominant condition called erythrokeratoderma variabilis, where again you get the Polyplanticratoderma, but you also get other more generalized hyperkeratosis, and you can also get erythemas, which can be transient or can last a few weeks. In addition, there is another missense mutation in connexin 31 called 66del-D, and this is associated with no skin disease, but with neuropathy and hearing loss. So let's try and understand a bit more about what these mutations are doing to connexin 31. So what we're showing you here is we've cloned the connexin 31 gene, and this is wild type connexin 31, and it reads through into green fluorescent protein, which is attached to which is linked in with the gene. So what happens when you transfect these cells? So we introduce this gene transiently into keratinocytes. You can see where the green protein is expressed, where the connexin protein is expressed, and you can see these are cell 1, cell 2. You can see there's aggregates of protein forming between the junction of the 2 cells, and this is forming a gap junction plaque. So one way to visualize this as well is you could inject this cell with a very small dye, and because the gap junction plaque is there, the dye will then move into the next cell. So you get cell communication. This is another way of visualizing the formation of the junction. Now, what about the skin disease mutations? Well, when we transfect them in, the cells do not form these gap junction plaques. They stay form aggregates within the Golgi. They're unable to form functional channels. So this is a mutation in these connexins are causing a trafficking defect in keratinocytes. The other thing we observed was that when we tried to grow patient cell lines, patient cells from biopsies from connexin 31 patients, they didn't like it. They died in the lab. And we see this sort of phenotype as well when we do the transfection studies. We can see that the cells are beginning to bleb and die. So the keratinocytes do not like overexpressing these skin disease mutations. And a way of visualizing this cell death is to add a small dye called propylium iodide into the culture media. And as the cells are dying, as the membrane's blebbing, the dye can then enter into the cell and go to the nucleus. So you can see how many cells are dying when they express this mutant connexin. So remember I mentioned this neuropathy and hearing loss mutation. So what's this mutation doing? Well, if you transfect it into keratinocytes, you do see some sort of trafficking defect, but very different to that of the skin disease mutations. The other striking thing is you don't see any increased cell death. So this suggests that these mutations are processed differently in keratinocytes. So what's happening with this mutation when you go into a neuronal type cell line? So in this case, we're using a cell line called SHSY5Y, which is a immortalized cell line from a neuroblastoma line. And you can induce neurite outgrowth when you add retinoic acid or brain-derived neurotrophic factor to the media. And what we wondered is what happens if we introduced wild-type connexin 31 and the mutant connexins into this cell line without adding the drugs to induce differentiation. And if you look in panel C here, this is transfecting the cells with wild type connexin 31. You can see where the protein is expressed, and you can see all these neurite outgrowths are forming, and you also see increased expression of neurofilament M protein as well. So this connexin is able to induce differentiation in the cell line. So would you see this with all connexins? Well, no. If you transfect the cells with connexin 30, for example, even though you get a nice gap junction plaque-like forming, the cells do not start to differentiation. So it's something very special about connexin 31's function. So what about the two skin disease mutations? Well, when you transfect these cells with R42P or C86S, they behave like wild type connexin 31. You get this induction of these neural outgrowths, which you can quantify. What about the neuropathy mutation, 66LD? This reduces very little, if any, neuronal differentiation. So what I'm showing you here is the fact that different mutations in the same gene, which cause different diseases, can be modeled in the lab, and you can see how different mutations have a different effect in cell lines. So as I mentioned earlier, the beauty about keratinocytes, as well as looking at them in monolayer, which I've been showing you just now, you can also form 3D models. And briefly, to do this, you seed your keratinocyte cells in culture media on a matrix of collagen with fibroblasts. You leave those cells for a little while, and then this is the key thing. You then lift the culture above to the air-liquid interface. And this induces keratinocyte differentiation. So after about 10 days to 2 weeks, you see the development of an epidermis, and you even get a reasonable stratum corneum as well. So this allows us to understand, dissect the function of Polyplanticratoderma genes in 3D modeling, in 3D models. So we can manipulate them. So going back to the connexin story, this is just wild type connexin 31. Again, it's tagged with the green fluorescent protein, but instead of doing a transient transfection, we're using a retrovirus so that all the keratinocytes are expressing wild type connexin 31. And you can see the expression of the protein in the cells, and you can see it's going to the membrane and it's forming what we assume are nice gap junction plaques. And then you do the differentiation, and you can then do some investigations of the histology or the molecular mechanisms which are associated with overexpressing connexin 31 in these cells. So as well as overexpressing, you can also do knockdown. So you can knock down particular genes to see how that affects differentiation. But as well as just looking at the histology, you can also then do some molecular investigations. So what I'm showing you here is if you knock down another gene associated with keratoderma called IROM2 in keratinocytes and then do 3D cultures, what you see when you don't knock it down is you see very relatively low expression of this keratin called keratin 6. But when you knock down IROM2, you get a dramatic increase in the expression of keratin 6. So it's clear that this protein, IROM2, is in some way molecular regulating keratin expression. But as well as just looking at candidate proteins, you can obviously apply omics. And so here we've applied RNA-seq and to see what happens when you knock down a particular gene in 3D culture. And there's a number of ways of analyzing this, but I'm just going to show you a pathway type analysis. And this is showing you that when you knock down that gene IROM2, what happens to which genes get upregulated. And you see it's genes that are associated with the cornified envelope and, for example, intermediate filaments. Keratin 6 is a member of the intermediate filament family. And then genes that come down are associated with other functions, including apoptosis and NF-kappa B signaling. So you can home into this data and understand a bit more about the signaling that's going on. So now I'm going to switch to animal models of keratodermas. And the best model out there is one that's been worked extensively on by Professor Pierre Coulomb. and it's where you knock out keratin 16. Mutations in keratin 16 are associated with Pacquiao congenita, which is a Polyplanticratoderma, and it's incredibly painful, like many other cratodermas. So when you knock this out in mice, you start to see this darkened hyperkeratotic skin thickening on the pads of the mouse's feet. And so this is really recapitulating what is happening in the patients. So a lot of studies are done in these mice to understand disease mechanisms that are going on. I just briefly want to mention some other mouse models, and these are and this stems from studying a family which had a recessive form of Polyplanticratoderma, and it's associated with exuberant scalp hair growth, so really thick, fast-growing hair. So there are spontaneous models out there, and this is a dog model which has a bit like the patients, has a homozygous missense mutation in the FAM83 gene, and you can see that the hair on the coat is very thick compared to its non-mutated control. And also you can see the formation of hyperkeratotic lesions on the foot pads on the dog's feet. The mice haven't looked at their feet, but they can see there clearly is a dramatic thickening of their coat hair. So, animal models out there can show some utility in understanding disease genes, palm-plantar disease genes. So I'm just sort of beginning to wrap up now, but so as I said at the beginning, there's probably 40 plus genes out there that can cause the Polyplanticratodermas. But are there some common pathways that we can perhaps home in on to target for therapeutic intervention? And this is just a little cartoon summarizing some of the things we know from doing molecular studies in the keratodermas. And what you can see is there clearly is a link with various signaling pathways, such as growth factor signaling pathways, the EGFR, and downstream MAP kinase signalings. There's a lot of desmosomal genes, aquaporin-5, iron-2, keratins, all coming in and targeting into this pathway here. So maybe these could be the type of pathways we should consider in developing new therapies. And indeed, some of these have been investigated already. So drugs like rapamycin and erlotinib, which is an EGFR inhibitor, have been used to show some efficacy in some of the keratoderma disorders like Pacquiao congenita and Olmsted syndrome. There is a whole wave of JAK-STAT inhibitors being developed at the moment, primarily for treating psoriasis and eczema, but I think some of these will also show great utility in the keratodermas. We and others have shown by studying mouse models or keratinocyte cell lines that oxidative stress seems to be one of the common pathways that we see in keratodermas. So maybe targeting antioxidant drugs may show some sort of therapeutic utility. Okay, that's all I'm going to say now, and I'll open the floor for questions.